Did you know that at this very moment, billions of creatures on Earth are not in control of their own bodies? Imagine a force that can rewrite behavior, hijack nervous systems, and turn free-willed animals into biological puppets, performing elaborate routines they would never choose, all to serve a parasite's reproductive cycle. Today, I'll reveal the 15 most terrifying examples of mind control in nature. Parasites so sophisticated, they don't just infect bodies. They reprogram brains, manipulate hormones, and orchestrate behaviors with precision that seems almost intelligent. These aren't simple infections. They're hostile takeovers of consciousness itself, proof that free will might be more fragile than we'd like to believe. From fungi that turn ants into zombies climbing to perfect death locations, to wasps that perform brain surgery on cockroaches, each represents millions of years of evolutionary refinement in the art of manipulation. Stay to the end, because number 1 is a parasite that infects an estimated 1/3 of all humans on Earth, subtly altering personality, risk-taking behavior, and potentially even psychiatric conditions. And you might be carrying it right now without knowing. Let's begin. Number 15. Hairworm, Spinochordodes tillinii. Picture a grasshopper on a hot summer day, behaving normally. Then suddenly, as night falls, something changes. The grasshopper becomes obsessively drawn to water, despite being a terrestrial insect that normally avoids drowning. It climbs the nearest plant overhanging a stream and throws itself in, committing suicide. From its convulsing body emerges a thin, dark worm, sometimes longer than the grasshopper itself. This is a hairworm, and for months it has been growing inside, waiting for this moment. Hairworms are aquatic parasites that must complete their life cycle in water, but spend their juvenile stage inside terrestrial insects. Research reveals that hairworm larvae produce proteins that alter the grasshopper's central nervous system, specifically targeting genes involved in light sensitivity and geotaxis. Infected grasshoppers become attracted to light, reflecting off water at night, and lose their fear of heights. The manipulation is so precise that infected grasshoppers will jump into swimming pools or artificial water sources. They're responding to visual cues of reflected light. The parasite has programmed a suicide algorithm: detect water reflection, override survival instinct, jump. The timing is equally sophisticated. The hairworm knows when it's ready and when external conditions support survival, adjusting manipulation timing accordingly. That's the hairworm, a parasite that teaches insects to love drowning, proof that suicide can be programmed. Number 14. Lancet liver fluke, Dicrocoelium dendriticum. Imagine an ant going about its colony duties during the day, but as evening approaches, something changes. Instead of returning to the nest, the ant climbs to the top of a grass blade, locks its mandibles onto the tip, and hangs there all night, exposed, vulnerable, waiting to be eaten by a grazing mammal. If it survives uneaten, sunrise releases it from this compulsion, and it returns to normal until the next evening. This is an ant infected by the lancet liver fluke. The fluke has a complex life cycle requiring 3 hosts: snails, ants, and grazing mammals. When a fluke larva infects an ant, one worm migrates to the ant's brain, specifically to the subesophageal ganglion, and insists there, becoming a permanent control implant. This brain worm produces compounds that override the ant's circadian rhythm. As evening temperatures drop, the infected ant compulsively climbs grass blades and locks its jaws, behavior called summit disease. positioning itself exactly where grazers crop vegetation. The brainworm doesn't actually reproduce. Only the flukes in the ant's abdomen mature if eaten. The brainworm sacrifices its own reproductive future to ensure its siblings' success, existing only to steer the ant toward its destination. It's reproductive altruism at the most disturbing level. That's the lancet liver fluke, a parasite that installs a sacrificial pilot in ant brains, proof that some organisms evolved specifically to be mind-control devices. Number 13. Jewel wasp, Ampulex compressa. Picture a wasp performing brain surgery with precision, inserting her stinger through specific weak points in a cockroach's exoskeleton, probing until she finds exact neurological targets, then injecting venom cocktails that turn the roach into a docile zombie that will walk willingly to its own tomb. This is the jewel wasp, iridescent and beautiful, performing nature's most sophisticated neuromodulation. The process unfolds in stages. First sting, the wasp strikes the cockroach's thoracic ganglion, temporarily paralyzing the front legs. Second sting, the wasp inserts her stinger into the roach's brain, probing around, essentially feeling for specific neural tissue. When she identifies the right location, she injects a venom cocktail containing dopamine and other neuromodulators. The effect is immediate and specific. The cockroach loses its escape reflex, but everything else remains intact. The roach can walk, groom, respond to touch. It's just lost all motivation to escape. The wasp then uses the roach's antennae as a leash, walking it to a burrow like leading a dog. Inside, she lays a single egg on the roach's abdomen, then leaves. The roach stands motionless for a week, while the wasp larva eats it alive, carefully consuming non-essential tissues first. The roach never struggles, never attempts escape. The venom blocks octopamine pathways, the insect equivalent of adrenaline, removing the roach's ability to initiate escape behaviors. The roach still processes information and could physically run, but the neurological trigger has been chemically disconnected. It's not paralyzed, it's unmotivated. That's the jewel wasp, beauty performing brain surgery, venom that removes will without removing ability. Number 12. Green-banded broodsac, Leucochloridium paradoxum. Imagine a snail's eyestalks swelling grotesquely, pulsating with thick, colorful bands of green and brown, throbbing rhythmically to look exactly like caterpillars. The snail, which should hide in darkness, instead climbs into open sunlight, where birds can see it. This is a snail infected by the green-banded broodsac, its eyestalks hijacked and transformed into parasite advertising. The broodsac must reproduce in bird intestines, but first develops in snails. Larval flukes invade the snail's eye stalks, filling them with hundreds of larvae arranged in colorful, pulsating segments. But physical invasion isn't enough. The parasite must get the snail eaten by a bird, requiring two behavioral modifications. First, the infected snail loses its photophobic response and seeks exposed locations in direct sunlight. Second, the broodsac makes the eye stalks pulsate rhythmically, mimicking caterpillar movement. Birds see these throbbing eye stalks and perceive them as prey. When a bird attacks, it typically pecks off only the eye stalks, allowing the snail to survive and regenerate its eye stalks over weeks. The broodsac can reinvade, creating an infected snail that can be harvested multiple times. The visual mimicry is sophisticated. The broodsac's patterns, colors, and pulsation frequency match caterpillars common in the snail's habitat. Different brood sac populations produce different patterns, suggesting evolutionary fine-tuning to local bird preferences. That's the green-banded brood sac, a parasite that turns eyestalks into advertisements, a creature that makes prey want to be seen. Number 11. Barnacles, Sacculina. Picture a crab behaving like a mother, carefully tending eggs tucked under its abdomen. Except this crab is male, and the eggs belong to a parasitic barnacle that has castrated the crab, feminized its behavior, and hijacked its entire reproductive system. This is Sacculina, perhaps the most invasive parasite in the ocean. Sacculina begins as a barnacle larva, but when it encounters a crab, it injects itself through the shell. Not the whole larva, but a few cells that grow root-like branches throughout the crab's body, infiltrating every organ, wrapping around the nervous system and endocrine glands. Eventually, a reproductive sac pushes out through the crab's abdomen, looking exactly like a crab egg mass. The crab treats these parasite eggs as its own. Male crabs develop female behavioral patterns, performing fanning and cleaning behaviors. The manipulation goes deeper. Sacculina castrates its host chemically, destroying reproductive organs while hijacking the neuroendocrine system. The crab pours all its energy into caring for parasite larvae, grooming them, protecting them, aerating them, even releasing them at optimal high tide. Sacculina isn't sitting inside the crab as a separate organism. It has become a distributed organ system, its tissues so intermingled with crab tissues that removal is impossible. The crab's body is no longer entirely its own. It's a chimera, part crab, part barnacle. That's Sacculina, a parasite that doesn't just infect, but integrates, transforming male into female, self into other. Number 10. Gordian worm, various species. Watch a praying mantis hunt with characteristic patience, a perfect predator. Then watch that same mantis throw itself into water and drown. From its body emerges a worm sometimes 3 times longer than the mantis itself, coiled like the legendary Gordian knot. This is a Gordian worm, orchestrating this suicide with precision. Gordian worms face the same challenge as hairworms. They're aquatic parasites using terrestrial hosts. In mantises, predators that actively avoid water, they must override deeply ingrained predatory instinct. Research reveals that Gordian worms produce proteins mimicking host neurotransmitters, altering gene expression that rewrites fundamental behaviors. But here's what makes them sophisticated. They don't manipulate continuously, only when ready to emerge. For months, the worm grows while the mantis behaves normally. Only when the worm reaches maturity, and only when environmental conditions are right, does it trigger water-seeking behavior. The mantis gets one suicidal impulse, perfectly timed. The worm can't see or sense the outside world directly, yet it somehow knows when conditions are optimal. This suggests the parasite is reading the host's sensory information, viewing the world through the mantis's eyes, and deciding when the time is right. That's the Gordian worm, a parasite that waits patiently inside predators, proof that manipulation can be delayed and deployed with strategic timing. Number 9. Ophiocordyceps, zombie ant fungus. In tropical forests, ants infected by Ophiocordyceps, fungi perform one final death ritual with choreographed precision. They leave their colonies, climb vegetation to exactly 25 centimeters above ground, select the underside of a leaf with specific temperature and humidity, bite down on the central vein with enough force to lock their jaws permanently, and die. Days later, a fungal stalk erupts from the ant's head, releasing spores positioned to rain down on ant trails below. This is zombie ant fungus, representing one of the most studied examples of behavioral manipulation. The precision is disturbing. Infected ants climb to exactly the right height, select locations with specific microclimates, and perform the death bite at specific times of day. Recent research reveals how, and it's not what scientists expected. The fungus doesn't invade the ant's brain. Instead, fungal cells infiltrate muscle tissue throughout the body, forming networks that control muscle fibers directly. The ant's brain remains intact while its body moves under fungal control. But the behavioral control goes beyond motor manipulation. Infected ants show altered circadian rhythms, modified pheromone responses, changed phototaxis and geotaxis, and precise selection of death locations. The death bite itself is remarkable. Ants bite down with approximately 20 times normal force, and the jaws lock so firmly that ants remain attached for months after death. What makes Ophiocordyceps unsettling is its specificity. Different fungal species target different ant species with different death behaviors. Each has evolved distinct manipulation protocols, suggesting millions of years of co-evolutionary refinement. That's Ophiocordyceps, a fungus that remote controls ant bodies while leaving brains intact, proof that consciousness and control can be separated. Number 8. Euhaplorchis californiensis, brain fluke. Picture a killifish behaving bizarrely, swimming at the surface in broad daylight, rolling and shimmying to attract attention, making itself as conspicuous as possible where birds hunt. This is a fish with a brain full of parasites. Literally, hundreds of larval flatworms burrowed into its brain tissue, and those parasites are steering it toward predators. Euhaplorchis requires two hosts. It reproduces in shorebird intestines, but must develop in fish brains. Unlike parasites that use surgical precision, Euhaplorchis uses brute force. Hundreds of cysts distributed throughout the brain, particularly in regions controlling motor coordination and threat response. Research comparing infected and uninfected fish shows dramatic differences. Normal killifish stay in deeper water, school with others, and avoid bright areas. Infected fish swim at the surface, separate from schools, perform spinning behaviors, and seem drawn to areas with high bird activity. When researchers place model predators near fish, uninfected fish flee, while infected fish approach curiously or ignore the threat. The manipulation isn't subtle, it's overwhelming. With hundreds of cysts creating mini lesions everywhere, normal behavioral integration breaks down. It's not that specific behaviors are reprogrammed. Normal function is disrupted in ways that coincidentally make the fish more vulnerable. That's Euhaplorchis, a parasite using quantity over quality, proof that brain damage can be adaptive if it serves parasitic goals. Number 7. Dinocampus coccinelli, ladybug wasp. Watch a ladybug standing perfectly still, protecting a cocoon beneath its body, the wasp larva that just burst from its abdomen. The ladybug will stand guard for a week, occasionally twitching to discourage predators, never eating, never moving. The ladybug is protecting its killer. Dinocampus lays eggs inside ladybugs, and the larvae develop internally. When ready to pupate, the larva chews out through the beetle's abdomen. Approximately 25% of ladybugs survive and remain standing over the cocoon, twitching their warning-colored wing covers. The twitching and coloration actively deter predators. Wasps developing under living ladybug guards have much higher survival rates. The ladybug receives no benefit. It's not protecting its own offspring. The wasp has programmed a dead man switch. Even after the larva is gone, the manipulation persists, keeping the host alive and guarding just long enough for the wasp to complete pupation. That's Dinocampus, a parasite that turns prey into protectors, a wasp that programs guardianship beyond its own departure. Number 6. Toxoplasma gondii, preliminary. Here we edge toward mammals, toward us. Toxoplasma gondii is a single-celled parasite that can infect any warm-blooded animal, but can only reproduce sexually in cat intestines. The challenge is getting from intermediate hosts, like rodents, into cats. The solution is to make infected rodents lose their fear of cats and even become attracted to cat urine. Normal rodents show strong innate aversion to cat odors. They freeze, avoid areas marked with cat urine. This fear is hardwired, evolutionary. Infected rodents show the opposite. They're attracted to cat urine, prefer areas marked by cats, and some studies suggest they find the smell sexually arousing. The parasite is crossing wires between fear circuits and mating circuits. Toxoplasma cysts preferentially form in the amygdala and areas associated with reward and sexual behavior. The parasite produces dopamine and affects other neurotransmitter systems, rewiring how the rodent brain processes specific stimuli. But here's what makes Toxoplasma particularly relevant. It infects humans. An estimated 30 to 50% of the global human population carries Toxoplasma, typically from undercooked meat, contaminated water, or cat feces. In most people, infection is asymptomatic, but the parasite forms cysts in the brain that persist for life. Studies suggest Toxoplasma infection in humans correlates with subtle personality changes, increased risk-taking behavior, slower reaction times, and possible links to schizophrenia and other psychiatric conditions. The effects are subtle, but statistically significant across large populations. That's Toxoplasma gondii, and we'll return to it at number 1 with the full disturbing implications. Number 5. Myrmeconema neotropicum. Picture an ant with a bright red abdomen that looks exactly like a ripe berry. Birds that normally avoid ants see this berry and pluck it from the ant's body, eating the abdomen while the ant, remarkably, survives with head and thorax intact. This is an ant infected by Myrmeconema, a nematode that has transformed the ant's appearance to mimic fruit. The manipulation is multi-layered. First, the nematode causes the ant's abdomen to swell and change color from black to translucent, allowing red nematode eggs inside to show through, creating a berry-like appearance. Second, the parasite weakens the connection between abdomen and body, making it easy for birds to pluck off. Third, the ant's behavior changes. Infected ants are sluggish, hold their abdomens high, and don't respond to alarm pheromones. When a bird eats the abdomen, it consumes thousands of nematode eggs, which pass through the bird's digestive system and are deposited in droppings far from the original colony. The ant, missing its abdomen but still alive, can survive for months. The visual mimicry is sophisticated. The red color, glossy surface, and size closely match ripe fruits in the forest. The nematode is hijacking the bird's visual system, exploiting the fact that birds search for red, spherical objects when foraging. That's Myrmeconema, a parasite that paints ants to look like fruit. a nematode that understands bird visual preferences. Number 4. Glyptapanteles, zombie caterpillar bodyguard. Picture a caterpillar behaving normally. Then dozens of wasp larvae simultaneously chew through its skin and emerge, dropping to the branch below to spin cocoons. The caterpillar, instead of recovering or dying, remains stationed above them, spinning silk over the cocoons, thrashing violently at approaching predators, serving as a zombie bodyguard for its killers. Glyptapanteles wasps inject multiple eggs into caterpillars, and the larvae develop internally. When ready to pupate, they emerge simultaneously, a process that should be fatal. Instead, approximately 50% of caterpillars survive and enter Gordian mode. They stop eating, stop moving, except to defend the cocoons. The bodyguarding is specific and effective. The caterpillar spins silk over cocoons and performs violent head-swinging at threats. Studies show that guarded cocoons have much higher survival rates. Not all larvae emerge. 1 or 2 remain inside the caterpillar, possibly still secreting manipulation chemicals, essentially sacrificing their own pupation to ensure siblings survive. That's Glyptapanteles, a parasite that programs post-emergence bodyguarding, proof that manipulation can continue after parasites have left. Number 3. Strepsiptera, twisted wing parasites. Imagine insects so radically modified by parasites that taxonomists initially couldn't determine what species the hosts were. The parasites had so thoroughly transformed host morphology and behavior that infected insects looked like entirely different species. This is the effect of Strepsiptera, twisted wing parasites that castrate their hosts and hijack their behavior so profoundly that infected insects become something between their original species and the parasite itself. Strepsiptera females spend their entire lives inside their hosts. The hosts are profoundly altered by infection, a syndrome called stylopization that includes physical feminization or masculinization, altered development, color changes, and dramatic behavioral modifications. Infected bees remain at flowers later in the day than uninfected bees, increasing their visibility to male Strepsiptera searching for mates. The parasite is using the host as a dating advertisement. Infected wasps lose aggressive nest defense behaviors. What makes Strepsiptera remarkable is the intimacy. The parasite's body is completely integrated with the host's body, sharing circulatory systems with tissues so intermingled they form a single chimeric organism. The host's endocrine system is hijacked completely. Some infected hosts become intersex, physically intermediate between male and female. The parasite is rewriting the host's developmental program. That's Strepsiptera, parasites that transform hosts into chimeras, proof that the boundary between self and parasite can become so blurred that host becomes questionable. Number 2. Spinochordodes tellini, advanced case. We began with hairworms causing grasshoppers to drown, but let's return with deeper understanding. Recent research reveals that infected grasshoppers don't just jump into any water. They preferentially select moving water over still water, and they're more likely to jump when the water surface is rippled rather than smooth. This suggests the hairworm is not just triggering seek water, but specifically programming seek suitable water. The distinction between standing water that might dry up and flowing water that remains stable. The grasshopper is performing environmental assessment on behalf of a parasite it doesn't know is inside it. Even more remarkably, the manipulation is dose-dependent. Grasshoppers infected with larger worms show stronger water-seeking behavior. This suggests the parasite is producing compounds proportional to its own body size, effectively knowing when it's large enough to survive outside the host. The proteins produced by the worm mimic host neurotransmitters so precisely that they bind to the same receptors but degrade more slowly, creating sustained activation. Normal signaling becomes pathological persistence, and normal attraction to light becomes obsessive water-seeking. That's advanced hairworm manipulation, not just suicide programming, but environmental optimization, a parasite that makes hosts perform complex assessments they don't understand. Number 1. Toxoplasma gondii, full picture. Now we return to the parasite that might be inside your brain right now. Toxoplasma gondii infects an estimated 2.5 billion humans, 1/3 of the global population. And while most infections are asymptomatic, emerging research suggests this microscopic parasite might be subtly influencing human behavior, personality, and mental health in ways we're only beginning to understand. In rodents, Toxoplasma infection causes dramatic behavioral changes. Infected mice and rats lose their innate fear of cat odors and become attracted to the smell of cat urine. The mechanism is specific. The parasite forms cysts in the amygdala and produces dopamine directly, rewiring how the brain processes cat-related stimuli. The manipulation is so specific that infected rodents still fear other predators. Only their response to cats is reversed. Now, consider humans. We can't measure fear of cat odor the same way, and we're not eaten by cats. So what would Toxoplasma do in human brains? The parasite still forms cysts in the same brain regions: amygdala, prefrontal cortex, areas involved in emotion, decision-making, and risk assessment. And it still produces dopamine and affects other neurotransmitter systems. Epidemiological studies have found correlations between Toxoplasma infection and various behavioral and psychiatric conditions. Infected individuals show, on average, increased risk-taking behavior, slower reaction times, and higher rates of traffic accidents. Infected men tend to score higher on measures of suspicion, jealousy, and rule-breaking. Infected women tend to score higher on measures of warmth, conscientiousness, and social behavior. More controversially, some studies suggest links between Toxoplasma infection and schizophrenia, bipolar disorder, suicide attempts, and even entrepreneurial behavior. The parasite is found at higher rates in the brains of people with schizophrenia compared to controls. People with schizophrenia are more likely to own cats or have extensive cat exposure in childhood. Correlation, not causation, but the pattern is consistent. Here's where it becomes deeply unsettling. We don't know if these effects are intentional manipulation or accidental byproducts. In rodents, the behavioral changes clearly benefit the parasite. But humans aren't typically eaten by cats, so human behavioral changes don't obviously serve parasitic reproduction. Either the changes are side effects of manipulation evolved for rodent hosts, or there's a benefit we don't understand. Perhaps increasing human risk-taking makes us more likely to engage in behaviors that expose us to cat feces. The most disturbing possibility is that we can't know if our behaviors, preferences, or decisions are fully our own or subtly influenced by a parasite in our brain. The changes are subtle, nothing like the dramatic fear reversal in mice, but subtle doesn't mean insignificant. Population-level effects that are small per individual can be large in aggregate. Some researchers have proposed that Toxoplasma might influence cultural differences in personality traits, noting that countries with higher infection rates tend to score differently on measures of individualism and risk-taking. This is highly speculative, but the point is, if a third of humanity is infected with a brain parasite that demonstrably alters behavior in other mammals, and if that parasite persists in human brains for decades, affecting neurotransmitter systems involved in personality and behavior, then it's not unreasonable to ask, to what extent is human behavior and culture influenced by parasitic manipulation? The ethical and philosophical implications are staggering. We prosecute people for crimes they commit. But what if behavioral choices are influenced by parasitic manipulation? We celebrate entrepreneurial risk-taking as personal achievement. But what if it's partially parasitic influence? We construct entire theories of human nature, personality, culture. But what if we're studying humans plus Toxoplasma rather than humans alone? That's Toxoplasma gondii, a parasite that might be shaping human civilization in ways we don't recognize, a microbe that blurs the line between us and not us, proof that consciousness, personality, and choice might be less purely ours than we'd like to believe. From Toxoplasma potentially influencing a third of humanity, to zombie ants climbing to precise death locations, to wasps performing brain surgery on cockroaches, these 15 parasites share a terrifying truth. Free will might be more fragile than we thought, and behavior we assume is volitional might be chemically scripted by creatures we don't even know we're carrying. What strikes me most is the precision. These aren't random effects or crude manipulations. Hairworms make grasshoppers jump into flowing water rather than still water. Ophiocordyceps makes ants climb to exactly 25 centimeters and bite at exactly the right locations. Lancet liver flukes release control at dawn and reimpose it at dusk. This is targeted, specific, sophisticated manipulation, requiring the parasite to sense environmental conditions, track timing, and execute complex behavioral programs with precision that seems almost intelligent. The mechanisms are chemical: neurotransmitter mimics, hormone disruptors, proteins that bind to specific receptors. But the outcomes are behavioral. The parasite rewrites the host's decision-making processes, creating behaviors that serve parasitic reproduction while often killing the host. It's the ultimate hostile takeover: invasion, infiltration, and replacement of the command structure while leaving the body intact and functional. Perhaps most disturbing is that we're only beginning to understand how common this is. For decades, scientists assumed parasites simply consumed host resources. The idea that parasites could manipulate behavior seemed too complex. Now we know it's everywhere. And we've probably only identified a fraction because many manipulations are subtle enough that we don't recognize them as manipulation. In humans, we're left with uncomfortable questions. How many psychiatric conditions have parasitic components we haven't identified? How many behavioral differences between individuals or populations are influenced by infectious agents? How much of what we think of as human nature is actually humans plus parasites? These 15 parasites are warnings and invitations. Warnings that behavior can be hijacked, that consciousness doesn't guarantee control, that the brain is vulnerable to chemical reprogramming, and invitations to question our assumptions about free will, to investigate the biological basis of behavior more deeply, to consider that some of what we are might not be entirely us. Because sometimes the decision to climb isn't yours. Sometimes the fear that should protect you has been reversed. Sometimes what you think is personal preference is parasitic manipulation. And sometimes, perhaps most disturbingly, you might never know the difference, because the manipulation is so subtle, so integrated, so much a part of your normal thought processes that it feels exactly like choice, exactly like you, even when it's serving something else's reproductive needs.